It was wonderful, really, to listen to all these talks today. And I'm really, really inspired, and I got very many ideas and very many impulses to work a little bit also in the field of joining the art and science together. But I'm sure that some of my students also got inspired to work hard in the lab after they heard so many wonderful scientific talks as well. I will tell you today a little bit about the molecule. You will also watch the movie later if you stay, about the molecule that actually make us, that molecule that determines who we are, and that determines also the type of a creativity we are going to have, or the things how are we going to perceive. And I will tell you the very exciting story of this molecule, and I have chosen it because DNA today is not only a molecule of life, but it has been used in nanotechnology to build new structures. And I will show you a little bit, and even people who are working with the DNA structures needed to invent their own language. How are they going to describe those structures? But let me start with the exciting thing about the DNA discovery. Let me just show you for all non-chemists as well here, but you've probably seen that. This is probably the most used image, chemical image or biological image also in the media. So DNA consists of two single strands which are joined by hydrogen bonding into the double helix. Basically, the formula of life is very simple. It consists of 5 bases, 2 sugars, and lots of phosphates. Phosphates are these groups together. And this is actually what makes us. This is really amazing. You know, even if you look at our exhibition, Molecular Aesthetics, Molecules That Change the World, if you look the statistics next to the molecules, what kind of atoms they consist of, you will notice that some of the most amazing molecules that that change the life of a people consist of a very few different atoms. And this is the case, indeed, with the DNA. So how did everything happen? It was a long story, and it involved lots of scientists, and I have chosen just a few of them here, but there are, of course, many, many more, and lots of PhD students and undergraduate students as well. So it started when Friedrich Michner studied actually white blood cells, and he have noticed, and this was the end of the 19th century, that there is a substance that can be found in all cells. And he called that a nuclein. And he also noticed that it contains phosphorus. At that time, it wasn't so easy to isolate something, and you couldn't give it to the NMR, or you couldn't get a spectra as we do today. But you need to find very nice ways of exploring actually what you have. And he have shown that there is a phosphorus in this substance. And then a very colorful personality, Phoebus Lavin, and some of the historians of science will probably be able to tell you a little bit more about that. He did studies on the proteins and nucleins, and he has identified the 4 bases on which the DNA, which actually occur in this substance, which is in the nucleus. And he also discovered that there are 2 types of sugars which are bound to these bases. And one is called deoxyribose, and one is the ribose, and therefore therefore it comes the name DNA and RNA. So DNA and RNA are similar types of the molecules, but they contain different sugars, and RNA has a different base as well. But actually, people have been thinking about how the life is happening, how the information is transferred. Why, if I have a green eye, if my mom had a green eyes, why do I get a green eyes as well? And of course, there was lots of research done in genetics from 17th century as well and earlier. But basically, what is the chemical basis of the life? And one of the interesting discoveries came from viruses. Viruses have been a very important tool for the discovery of many things in biology. And Stanley, he worked on tobacco mosaic virus, which you showed, could see in many lectures, has an interesting structure as well. And he have noticed that there is an RNA in the center of a virus, and if he changes this RNA, he changes the property of a viral shell. So he concluded that there must be something in this RNA which causes the change of information, biological information. And then the Hershey did some experiments of bacteriophage. This is this funny looking virus which attacks the plants, and it has the DNA in the middle. And he has noticed that if he takes the DNA out of the virus, nothing happened to the cell. But if there is a DNA in the bacteriophage, something will happen in the affected cell, and the affected cell will produce more viruses and die at the end. And then, you know, all these informations came together, and Linus Pauling, which was one of the greatest chemists, he was also puzzled by a structure of this molecule, which is so present in the living organisms, and he actually speculated what kind of structure this could be, and he falsely suggested a triple helix. But there were these guys. I will not go into the whole discovery of a structure or explanation of a structure is a little bit of a scientific thriller, and I hope you will stay for a movie and you can see how many people were involved in all of this. But these two guys, Francis Crick and James Watson, they were in Cambridge and in 1953, and they were actually not biologists or biochemists or even experimental chemists. And then they listened to some of the lectures of the other people who were at that time in the Cambridge, and they were sitting, there is a famous pub in Cambridge as well where you can go and visit, and they were sitting there and they were playing around because they had some data, crystallographic data. That means they got the data which were collected, and they played around, and this is the original sketch. They made a sketch of a DNA out of the data they had, and they suggested that this must be a double helix. And they said that it was so beautiful as well that this seemed to be like the only solution that could happen. And one of the great stuff, you know, sometimes you have, as a scientist, you need to publish your data and you have to present it and you have to be very open to critique as well. And what is beautiful about this paper, this was a paper in Nature, and you have heard this name Nature so often. It's a one-page paper where they describe how did they come up with a structure. And what is the most interesting, and I made this a little bit bigger, is they say in one part of the paper, it has not escaped our notice that a specific pairing we have postulated, that means the pairing of a double helix, suggests a possible copying mechanism for the genetic material. And this sentence has caused the whole revolution. Okay, we wouldn't have genetics, we wouldn't have all of these things that we are debating, we wouldn't have so many political controversies about the genetically modified food and all the stuff that we have, if this sentence didn't actually give the hint, and other scientists would have gotten the hint later, of course, that there is something very special about the double helix that enables the transformation of the information. I will not go into the details about all the things that, you know, how the transformation is transferred, but just for you to understand how DNA is a very complex molecule. Actually, it looks very simple, but there is a large number of proteins that is involved in making this copying mechanism, in transferring genetic information. And this is just the one image that I like to show always. We have this double helix DNA, a wonderful molecule. Humans have, and you know, double helix is formed, as I told you, because the bases are having these weak hydrogen bondings in between. And humans have around 3 billion base pairs. That's a huge number. That's a huge molecule. And these 3 billion base pairs are packed into 20 to 25,000 genes. And the genes are the little packages of information that can be transferred further. Okay? And I like to do some statistical. I always look and try to calculate and try to use the symbols. How am I going to explain what is that happening? And if you look within yourself, human DNA is actually in the single cell. It's 3 meters long if you will put it in a linear way. That means if we will be able to take the DNA out of our cells, and we are, and we will stretch it, it will be around 3 meters long. That's a huge distance. And if you would calculate how many cells we have, you will come that we have altogether 2 times 10 on 13 meters of DNA in our body. So here is the number of DNA walking around. So basically, if you stretch this, you could have 67 trips from the Earth to Sun and back. Just imagine how big this number is. So much of this molecule you have in yourself. We should definitely be very happy that we exist and that we are here. And whenever you have a problem or you feel a little bit down, just think, you have lots of beautiful molecules in yourself. I actually talked to one of my students. He miscalculated this number, and he said, I think something like 70 trips to moon and back, and then he ran to me in panic and he said, I miscalculated, we have much more. And you know, even for us who are working with this molecule, it's always amazing when we actually try to compare the numbers and put them in the perspective. But how is it possible that so much, that 3 meters of a DNA comes into the cell, which is around 1 micrometer, even a little bit bigger? DNA needs to get wrapped. And I want to show you, why am I showing this? I don't want to discuss all the details. I just want to show you how the molecules can fit beautifully together to make a functional, act functionally and create novel structures. So in DNA wrapping, there is a particular protein, which is a very round protein involved, and then the DNA is basically wrapping in the coils around this protein. So you start with a double helix, and then you have a coiling, and this happens many, many, many times, and finally you end up with this. Maybe you have seen that. This is a chromosome, and we have more than 40 of them. Okay, so basically 3 meters of a DNA are wrapped around this protein to be packed into the DNA. And the information, just shortly how the information is transferred, of course there are plenty of proteins and the enzymes which are involved in the transformation of your genetic information. That means DNA controls the expression of the proteins. It controls the whole cell machinery. Whenever you have a desire to drink the coffee, there is something in the morning, there is something telling you need to drink this coffee in the morning. Whenever you have something to eat, you need to have the range of enzymes who will actually transform this food into the energy. You have to have a cellular mechanism that are going to detoxify your cells, all kinds of rubbish that is formed as a side product. And the DNA is basically controlling all of that on the daily basis, in the milliseconds as well. And how this happens, basically you have to unwrap this double helix and you need to create a single strand, which is then copied. If you have, and this, of course, there are also the rules how to copy this, because only particular bases will recognize a particular base. So for example, A will recognize the T, or in this case, U in an RNA. C will pair with a G. So this is the base pairing. And basically there are 2 steps in the information. You have a first transcription. When you have a single-stranded DNA, it's transcribed, copied into messenger RNA. And the difference is, this is the similar single strand, and you have a same number of bases. It's just that the T is changed into the U. And when you have this messenger RNA, it will transfer the information in the process called translation with the help of lots of particularly special machineries, and then the proteins can be synthesized. And this is basically a code language. The DNA is it's a mix of codes. It's a code language which is based, and Joe Davis told you also about that. So 3 bases will basically code for a single building block of the protein. And then you have also the codes for stopping the synthesis of the protein or starting the synthesis of the protein. But what I wanted to show you basically, that when the messenger RNA gets a particular information, and this information needs to be transferred into the protein, there is a very peculiar machinery that is doing that, and these little machines are called, they're actually big machines compared to the rest of the things in the cells, they are called ribosomes. And the story of ribosome is an interesting story, because it took more than 30 years basically to get the structure of the ribosome. And I had a chance to meet this lady. The Nobel Prize for ribosome machinery was awarded in 2009, and she told me that she worked 20 years in obtaining the crystals, you know, beautiful crystals that Rab told you, that sometimes, you know, you come in the morning, you don't have anything in the flask. Well, it took her and her team 20 years to get the crystals. They went even so far that they tried to do the crystallization in space. So they put the crystals, they put the material, they isolated the ribosome, they had an agreement with NASA, they put it into the shuttle, and the crystal have been growing in the space. But what happened, the shuttle was falling somewhere in Siberia, and everything happened fine, the crystals were still undamaged, but when the crew was taking it out, they dropped the flasks with the crystals. So the ribosome crystals were basically lost somewhere in Siberia. But they didn't give up. They didn't give up, and they got a crystal structure. But getting a crystal structure of a biomolecule is not the end of the story. You have the structure. But how are you going to explain how does it work? So you need to be a little bit of a genius to put all the pieces of the puzzle together and do lots of detective work to actually discover how it works. And she gave me this movie, and this movie is now also available on YouTube. So that means this is a very popular molecule to look like. I just want to show you. I will click that. I checked that before, and this worked. Let's try it. Oh, we will not give up. Okay, it's coming. This is just the credits to the Ada Jonat works at the Weizmann Institute, but she did the work in Germany as well. I just wanted to show you, this is just for the, it's a really beautiful movie. So the messenger RNA comes with the information about the synthesis of your protein onto the ribosomal unit. Then two subunits will actually come and join because they realize there is a signal that says, okay, there is something happening, we need to start the synthesis. They will join together, okay? And look at this, on one side, the RNA is coming in, and the ribosome is reading it, and then the building blocks, the other RNA, which brings the building blocks, comes from one side, the ribosome is taking it, throwing away the RNA that doesn't need, and down there, the protein will be synthesized. Okay, this is just magic. When I see that, I feel, okay, now I understand why I got into chemistry and science. So this is happening in our bodies constantly, because constantly you have to produce the things. So basically, information comes, it's read here, then the building blocks are coming in with and then the protein is synthesized over there. When the synthesis, you know, you have to say to the machine, when are you going to stop? You need to stop it somewhere, otherwise it's going to be exhausted, and then at the end you don't have enough fuel. And what happens, suddenly the end codon on the messenger RNA came, the building block that came in said, okay, now we finished with the synthesis of the protein, now we don't need you anymore, ribosome, and then the little subunits that join the ribosome at the beginning go away. And the rest of the building blocks which are not needed are then degraded, or they are just expelled out of the cell. But this is, for example, a wonderful, wonderful example where the crystal structure actually led to the discovery of the whole principle. Of course, there are lots of people who have been working and suggesting the theories, how the whole information and the protein synthesis happens. But once when you can make these movies, then you can really grasp what is happening in our bodies. It's always amazing to see. The other question that I would just like to tell you a little bit about when we are talking about the DNA, how does the cell know which genes need to be switched on? How does it know that if you drank too much alcohol, a particular enzyme needs to be synthesized? It's called alcohol dehydrogenase. Something must be happening in the DNA. Something needs to give a signal to the DNA, okay, I had too much drink, Let's make some alcohol dehydrogenase. And this is a big mystery. This is something, that's why I said in my abstract that we have just scratched the surface. We know so much, but we don't know basically very much about the very important things as well. So there are several theories that these little histones, which I showed you at the beginning, you know, that are wrapping the DNA, there are theories that they play amazingly big role, because for the DNA to be read out, it needs to be unwrapped, it needs to be stretched. Okay, it can't be read when it's still basically wrapped around the histones. So this might be playing a huge, huge role. And then there is a protein which destroys anything bad in our cell, basically, and signals to the cell that something is dead and needs to get out. So this is probably very important. And in 2008, the scientists also discovered that there are some particular activator proteins which are also responsible for switching on the genes. And the question is this protein machinery which is in our cells, which I showed you partially as well, is that the only thing that is responsible for a transfer of genetic information or switching on the gene? And probably not, because there is a new field, and I just wanted to mention that because this is a word that you will be hearing also in the newspaper and in the media, and it's important to know what is that about. There is a new field which is called epigenetics, and this basically says, and I put this picture because I had a picture of female twins, and then I thought I need to put the male twins as well to be politically correct. Well, epigenetics says that hereditary changes, that means something that I inherited from my parents and something that my children are going to inherit, is not only a function of a DNA sequence. That means all the information is not only in the DNA sequence, but what matters is also what I'm doing with my life throughout my life. That means all the small molecules that I'm taking on, the things that I eat, the air that I breathe. That sounds very poetic. This all can play a role in the switching on the genes. And how did they come to? The scientists basically did lots of work with the twins because they have genetically the same type of a basis in the DNA. Okay? However, they still have different illnesses, even if they live in the similar environments, and in particular, if they are separated at some point of the life, they will suffer from the different illnesses. And if the DNA is the only thing that controls our information, that shouldn't probably be happening. And so there is really this field of epigenetics picked up in the last 5 years. And chemicals or any other stress factors in our environment will influence the gene changes. And recently there was a paper where they did studies on the flies as well. So they would change the type of you know, you can genetically modify the flies so they have a different color of the eyes, for example. And so they will expose these flies to a certain kind of stress, and they will see that suddenly the fly gets the green eyes, and these eyes are transferred to the next generations. That means whatever I do to my genes today, it will really happen that even my children or grandchildren might suffer from this kind of illnesses. There are really wonderful papers out there that I recommend you to read. It's even interesting to read for non-scientists, which is quite nice. So I just wanted to point out that there are still many mysteries to be solved when we are talking about the biology of a DNA. Just where is the DNA coming from? I think I will skip that because I was planning, I will not have enough time to talk about this. You know, scientists are always asking the question. So we have a DNA, but where is the DNA coming from? Where was the first DNA made? How was the first DNA made? And so, to cut the long story short, there are theories out there that DNA was not the first molecule that transferred the information. Actually, RNA was. And if you remember, RNA has only single strand. This is not a double helix. And instead of T base, it has a U base. And it's believed that because RNA is basically responsible for manufacture of proteins, and it has also properties to influence a certain synthesis. So it's a kind of catalyst, something that Raf told you about some chemicals or some molecules can be used to synthesize other molecules. And therefore people believe that RNA was actually the first molecule that appeared, because RNA replication system is simpler. There are few proteins that are involved in the transfer of information. DNA nucleotides, they were also, these building blocks, they were also shown that they can be made from RNA nucleotides. And then there are some other points which point out that the RNA was the first one. And in 2009, I'm putting these years here because I want to show you, although we know the DNA structure from 1953, I mean, there are constantly really important information being produced in the labs all over the world. So in 2009, the chemists, actually British chemists, they have managed to produce the RNA base from the very simple building blocks which we believe existed in primordial soup. That means when the earth was still not very friendly place to live, there were very simple molecules floating around, and they have shown that if you take these simple molecules that were floating around and you use the light and the water, you can basically make, this was a wonderful paper, you can make an RNA base. And this was then one more step into the proving the theory that RNA came first, then the DNA was synthesized. Why did DNA took over the control of the cells? Why don't we have our RNA in our bodies? This is also a good question because it appears before. And there is a very important difference between these two molecules, and this is particularly important also for the nanotechnological purposes. RNA is more flexible because it's a single strand. It is a catalyst. That means it can be involved in the production of new molecules. And it has a very simple mechanism of replication. But DNA is more chemically stable. It's a double helix. It is stabilized with a special bonds. And it can form longer structures. Because of this double helix structure, it can form longer structures. And therefore, it can contain more information. And this is what is needed if you want complex life forms to be formed. Okay, so the DNA took over. There is also a group of scientists, and there is also another theory that viruses were crucial in this process, because the viruses, there is in the change, you know, now with the DNA analysis we can actually trace the organisms and we can show which organisms are basically related. So what people have found is that the bacteria, which are single cell organisms, and eukaryotes, which are multi-cell organisms, they have certain proteins which are more similar to viruses than to each other, which means that some of the theories says that somewhere at the point of a life development, viruses have been containing DNA, and then the life branched into two directions, into one-cell organisms and into multi-cell organisms as well, with a particular complex protein structures. So maybe the viruses are beginning of it all. You know, maybe we are really unfair to these viruses. So there are still, this story is to be continued, you know, so this is more like an inspirational thing for anybody who would like to get into the field. There are still many mysteries to be answered, okay? And some more mysteries, if anybody is interested in getting into the field of DNA research, the question is, when did DNA took over the control of the cell, and why? Why are we interested in this? Because we are curious. And because we are curious and we want to understand how the things happened, we would like to know that as well. And hopefully that will open some new areas of knowledge as well for us. How is the gene readout controlled and what affects it? As I told you, you know, now we are understanding a little bit more that there are so many proteins, there are so many molecules involved in the whole gene readout, but how is that controlled? If we know that, then we could control some of the illnesses, we could cure some of the genetic diseases, and we could basically understand much more what is happening within our genes. Today, we do have tools with which we can silence certain genes as well. And scientists have been using that to create special mouse models or special animal models. They will silence this particular gene. So for example, you create albino mice, for example, because you switch off the gene which is responsible for fur color. So we have some of the tools, but the question is also which genes do we need to switch on or switch off in the case of the certain illnesses. And we still don't have this information. We do have information, we have a genome map of humans, but we still don't know how these genes are basically related to anything that is happening in our cells. We are just scratching the surface with that. So these are all interesting topics for anybody who wants to continue their career in science. I'm just jumping now a little bit further, so I will not stay into this biological part of the DNA. I want to tell you, DNA is synthesized in our body, and the real revolution into understanding the DNA came when we were capable of making the DNA chemically in the lab. And the guy who did that was Gobin Khorana. He was at the University of Wisconsin in the 60s, and he also got a Nobel Prize for his synthesis in 1968. And what he managed to do, he would take the building blocks, and then he would build a single-stranded DNA out of this. And he managed to make 77 base pairs. We have 3 billion of them in us, and he made 77 base pairs. It took him 20 years to make them. We do have a synthetic DNA now in the labs, and students don't need 20 years any longer to make the DNA because we have machines that make us for us. So this is a DNA synthesizer. This is how we chemists make it today. So basically it's just a machine. You can put a solution of your bases here. You have some particular chemicals which you need to do the chemical synthesis. Here you can write your sequence. And then you program your machine, you leave it overnight, depending on the size, and tomorrow you get a DNA. However, the limits of this machine is that you can synthesize only a particular number of base pairs. Okay, so I think today we can go up to 80, but so if you want a longer DNA, you have to find some biological ways of gluing it together, and people have been doing this. There were scientists who actually managed to make a Spanish flu virus DNA just a few years ago, and that caused lots of controversy because what could happen if any lab in the world could make some synthetically some potentially dangerous DNA? It's not so easy as some media would like to tell, so don't be afraid. It's not very easy to make that. So when we have a DNA, we actually need to know how many base pairs we have. So we use this gel electrophoresis, it's called, where you get these bands here, and they will tell you this is the DNA. If you have a particular marker here on the side, you can really say how much base pairs you have. Okay, so this is the way how you can very quickly then identify your DNA. And why is that important? I told you that this brought a kind of revolution in the science, because DNA is today not only the molecule of a life, and therefore the title of my talk, The Double Life of a Double Helix. It is also a building block in nanotechnology. And I will show you how this looks like. Once again, just to remind you, because sometimes, I can talk too much and then people get lost. This is the structure of a DNA. Joe Davis already mentioned that there are actually more structures which can be found. This Z-DNA was actually crystallized for the first time in 2005. That means also very recently we got a crystal structure of this DNA. And so I told you also that DNA is a stable structure. Double helix is a stable structure. But it's also a very flexible structure because it can be packed, it can be wrapped. Basically, we wrap these 3 meters of a DNA into our cells. Short double-stranded DNAs, that means short up to 50 base pairs, they're really rigid and straight. Okay, so this is the one of the differences and very important points for the building blocks in nanotechnology. Single-stranded DNA, that means when you just have one strand, is very flexible, okay? But the double-stranded is very rigid. And combination, if you combine both of them, if you combine this double-stranded rigid parts and a single-stranded DNA, you can have artificial DNA motifs. Okay, you can create new kinds of DNA, not now only the double helixes, you can put these double helixes into the new structures. The advantages of this is that you can, when you create these structures, you have been taking biological molecules to make that. That means you are not introducing some kind of inorganic compounds or some man-made compounds, which means that you have some biological properties probably of these structures. You can precisely design the structures as well, and then you can use both the chemistry, because you can synthesize up to 80 base pairs in the lab, and the biology to make particular structures as well. And here's the special language that the DNA nanotechnologies developed, because if you would like to draw, you know, we were talking so much about molecular aesthetics and representation. Can you imagine if you would need to draw these double helix for 200 base pairs? It will be very difficult. And then if you would add some particular structures here, it will be even more difficult. So we needed to learn how to simplify that. So basically the double helix is just represented by this ladder. And what can you have when you design the DNA as well? You can have a different kind of shapes that you can introduce. And for example, in A, you can see this normal double helical section of a DNA. But then you can have B, sticky ends. Nice name, it's a sticky end. It is basically a small number of bases at the end of the DNA, and you can stick it with a longer DNA which is complementary to these bases, therefore the sticky ends. You can have a bulge loops, you can have a junctions and crossovers. And the guy who was actually a pioneer, or I like to call him a father of this field, because lots of his students became professors and they work in this field as well. So, you know, he has a really network of his students who are really active in the field, is Ned Seaman from a New York University, because he told us the story once. He was in New York and he was very unhappy with his job because none of the students wanted to work with him because he had these crazy ideas. He would make crystals out of the DNA. I mean, people were really interested in making crystals out of proteins and ribosomes at that time. So he didn't have any students really. So he used to spend lots of time reading in the pubs. So he went to the pub and he said that he saw the painting of one of the artists that one of the young artists that did the exhibit in the pub, and he saw this artist putting a different symmetries, this kind of symmetries together. That means the crosses with a different kind of colors, and he was creating all these wonderful paintings. And then it occurred to Ned Seaman that while he was thinking about the DNA, that he has a tool where he can create new structures out of the DNA, that he can create. Why do we always think about the DNA as a double helix? We can use and create new kind of structures. And exactly here, he then postulated a set of rules. How can you make these structures? For example, he also said that there are particularly chemical grooves that you can add at the ends. And he has also designed a computer program which was called the Sequin, where you can basically put the sequences in the computer, or you can put the designs that you would like to have, and the computer will calculate what kind of base pairs do you need to make this design. There are also particular rules. If you want to make something more flexible, you will put many T bases around here. It's a whole new world of a nanostructure design. I will not go so much into the details of that as well. So for example, just to show you, you can make lots of branch structures that can be the basis of your architectural buildings. You can introduce lots of symmetries. Now the sticky ends are very important because this is how you are basically sticking the pieces together. You can never make a big DNA and then just, actually you can make, I will tell you about this, I should not jump to the conclusions. You can have a big DNA and fold it. But let's talk about sticky ends. So you could basically, just for you to understand, DNA helix works on this complementarity of a certain basis. So if you have a CACG on one side, then you can basically bind it with if you have a complementary basis on the other side, and you are sticking the things together with a hydrogen bonding, which is a kind of not a strongest bonding, and it is a basis of a supramolecular chemistry basically. If you want to make a longer DNA, or if you want to then not only keep the DNA together by this weakly hydrogen bonding, you can use the ligation. Ligation basically involves the use of the protein, and you can get rid of these little holes in there. So you are creating the smooth strands. And what can you do with this as well? For example, if you are taking one tile and you would like to stick it, you can, if you design your sequences correctly, you can make a bigger and bigger and bigger structures. And then you can introduce on these corners, you can put the DNA with a particular chemical functionality. So basically you can attach something onto the surface of a DNA. And this is how this would look like. And of course, you have to see what you have made, and this is not always the easiest thing to do, so people are using AFM as a technology to discover this. So basically you can see a beautiful tiles of a DNA here. And these white spots are basically some proteins which are sticking out of it. So these are the false colors, and we were talking about this, how you color that stuff. So, you know, you create some rigid and flat building blocks and then you can put them together to create more structures. So, you know, there were railroad tracks which were created, and then you can go not from flat, you can go into the dimensions as well. So you have a two-dimensional lattices, and Cheng De Mao is one of the students of Ned Seaman, who's really one of the biggest experts in the field of nanotechnology. And you can go on, you can build even more complex structures. It does require a little bit of computer power and a lot of knowledge and creativity as well. So people have been creating this kind of structures, and you can see these triangles here as well. Or you can create some kind of beehive structures as well. You can create smileys, but I will come to that. But first of all, you can do all kinds of different shapes. And you know, the question is now again, okay, why do we actually want to do them? Is that only for the sake of showing that we can do them? And yes, of course, at the beginning of the field development, although Ned Seaman had a vision, and I will tell you about his vision, because his vision came true just last year. He was also interested to see what are the limits of these structural building blocks. So yes, at the beginning it was about building new structures and trying out what kind of structures we can make. Yeah, I just wanted to tell you that because the structures became more and more complex, the scientists decided that they will introduce also particular code names for particular structures and the coloring of the structures. So basically, when you look at the DNA nanostructuring papers, you might see lots of these schemes. I can explain you later in the discussion time, or when we have a dinner, what that means. This will take a little bit long now. So the question was, why are we making this? Because we would like to do a self-assembly, and probably some of you who heard some talks already, self-assembly is a very important topic in the chemistry, in the biology. We are held together because our parts of our cells are self-assembling, our cells are self-assembling, and we still understand a little of it. How does it all happen? And of course, you know, to create different kind of functional devices, for example, some kind of chips, you also want to self-assemble particular devices together. In this case, people have been using gold nanoparticles. So this is really a synthesis of DNA nanotechnology and the nanotechnology that people usually relate to gold nanoparticles together. So basically you can, by a careful design, you can immobilize these gold nanoparticles in a particular positions. So you basically control a little bit of a self-assembly of this system. You can also put some proteins together on the DNA. And why is that important? Because we want to know how proteins interact together. We know that they work in synergy, that sometimes they don't like each other, but we still don't understand why is that happening. If you would have a chance to control the distance between two proteins and put them close together, then you can study how they are interacting, what is happening if you are adding something to them. And DNA enables that because you can basically control the distances in these tiles. So you can control the distances between these two elements as well. I told you at the beginning that Nate Seaman has used, of course, chemically synthesized DNA, lots of many strands. And I need to tell you that this kind of science is not cheap, because to create certain structures, you need sometimes hundreds of small strands, and this is not, it doesn't come cheap, I can tell you. But then, just recently, a few years ago, there was a guy who actually took a natural DNA. He took the DNA from the virus, and this is a single-stranded viruses. I forgot to tell you there are viruses that have a double helix, but they have also a single-stranded DNA. And he took the DNA out of this virus. Then he wrote the program, and he got out the sequences of a small DNA helper strands, he called them, that help him fold this long DNA into different shapes. So basically you can program what kind of shapes you would like to have. And this smiley appeared on the cover of a Nature because, of course, it is impressive. So what happens, you take a natural DNA, which is very long, and then with the help of a smaller synthetically available DNAs and with the help of a computer, you can mix them all together. You let them hybridize a little bit, and then you get this kind of forms. Okay, so you can get smileys, you can get stars, you can get the triangles. It always looks so simple. You mix one DNA with many other DNAs together, you let them, hybridize. It's never so easy. So it takes lots of times to get these images. Of course, it's much easier right now because people know some tricks. So this is called DNA origami, because you are basically taking, as in Japanese art, you are taking a piece of paper and you are folding it into the different shapes. In this case, you are taking a piece of DNA and you are folding it with the help of other DNAs into the different shapes as well. So this is a DNA origami, and this is what Tammy Spector actually showed you, and I was so happy that she showed you that. You can also make 3D structures, and this is the example of a DNA cage. It also takes a little bit of a design, but what is particular about this DNA cage is that it has a lock and a key. And the lock and the key are also a DNA strands. So basically you can close the cage and you can open the cage. Isn't that beautiful? And you can basically see that also with microscopy. This doesn't have any application yet, apart of being a very interesting system. But you could imagine that if you create a box big enough or small enough, you could basically contain a particular drug or a particular molecule in the box and open it as you would like when it's necessary. Maybe introduce it to the cell and then open it when the cell needs it. Or you can maybe use it for storage of a particular materials. Okay, so 3D structures are also possible to make. So we start with this flat, rigid tiles, and then we were building the 2D structures. Now 3D structures are also possible to make, and for example, something like this. And how do we know that we have them? In this case, they were using transmission electron microscopy. So you put the gold nanoparticles. This case is a little bit more interesting. You put the gold nanoparticles that are spheric and of a nano size, of a different shape onto this DNA structures, and what you actually get on the TEM image, you get this really regular assembly of a bigger and a smaller nanoparticles. So basically now we are doing a self-assembly of nanoparticles onto the surfaces. I put one of my students here because he drew such a nice scheme as well, just to show you again DNA origami once again. How does it work? So you have a single strand, you have lots of helper strands, and then they're folding the single strand into the particular shape. And here, one of my students, he tried to do the hat. He managed. The hat was placed on the head of a smiley. This is the work of my ex-boss, so I'm very happy to show you that. So, you know, scientists can have a sense of humor, and they like to have fun as well. Yes, and now the vision comes, the vision of Annette Seaman, why he actually started working on these nanostructures and created a whole new field. He wanted to use the DNA. He started as a crystallographer, and he was doing a crystallography over proteins. And, you know, the people who are working with proteins, they will know it's not easy to crystallize them. And if you want to know the structure of a protein, you need to have a crystal, you need to have an X-ray. And he thought, maybe I can make a DNA cage. Maybe I can make a 3D structure, because DNA is crystallizing quite nicely, where in the middle I can put a protein. And then the DNA crystallization is going to force this protein to crystallize as well. So he has made in 2010, and this was just in 2009, exactly, 2009, and he actually came to Karlsruhe, and we had a summer school here, and he presented this work fresh over the press, so we were all excited. What he has made, you need to look at the structure a little bit more. Basically, his team designed a DNA nanostructure, and when you look, the symmetry of this structure, there's a cube in the middle. You see the cube? If you look carefully, it's a little bit difficult to see with all the coloring. So you have a cube, and they managed to crystallize that. The next step they are working on is basically to create this structure together with a protein that would fit exactly the dimensions, because you can design these cubes to fit dimensions of the proteins that you would like to crystallize. So you can basically put the protein in the middle, and then you could crystallize it and you could know the structure. So these DNA structures, they can really be tools to help us to move the field forward. And one funny thing, which I like to show, he told us in a lecture 2009, how hard it is to make this and how long did it take to make that. And he said, time in years, it equal dimensions square. That means if you want to make three-dimensional nanostructure, that will take you, how much? 27 years to do that. It took him around 27 years from the concept to making a 3D structure. And now, of course, it's getting easier and easier to make that. But you know, you have to appreciate the love for the science he has, and you have to appreciate his creativity and patience as well. And when you see his students, you also see that he was able to transfer this knowledge as well to the other students. So sometimes it takes a long time. Sometimes you need to believe into yourself a lot, because nobody else believes that your work is worth doing, particularly if you're doing something really new. But if you believe in it and you have enough time in your hands and a little bit of funding from your government, then you can create really wonderful structures. And now I'm over the time, but I will just quickly go through some more applications, because I told you these nanostructures, they can be useful, and we are just discovering how to make them useful. So they can be useful for crystallizing the things. They can be useful to make diagnostic chips. Today, of course, we would like to have more personalized medicine. That means if you come to the doctor, you would like to have your blood samples taken, and you would like to know what kind of illnesses you have, what kind of allergies you have, what kind of drugs can be prescribed to you. And this requires a design of a certain kind of chips. That means you have a small area where you can put many different molecules that will interact with the molecules in your body, and then you get a signal, okay, this person can be allergic to penicillin or something, in a quick way. It's a little bit more complex than this, but you can use the DNA and this ability to build the hydrogen bonding and create a double helix to actually make a diagnostic chips, both for a DNA and for a different proteins. You can create artificial proteins. And why do we want to create artificial proteins when we don't even know and understand what is the function of our natural proteins? Well, you know, what Professor Linder says, it's a challenge, and the challenge, it's related also, the creativity is needed, But of course, when you are creating a new artificial enzymes, every time you understand a little bit more about the enzymes that you have or the proteins you have. So we have made here a natural protein, it's myoglobin, you heard that already. We couple it to the DNA, we couple it to the particular groups, and we make it light sensitive. So this protein, which is not natural light sensitive, started doing its functions only when it was irradiated with light. So this is one way. You can make biosensing. Biosensing sounds really terrible, but you know, it's not terroristics. It's basically you want to explore the interactions within your body. And this is, for example, a colorimetric test based on the DNA. You want to see, for example, if you have a particular DNA which is responsible for a particular type of the cancer, a very fast test is developed. So if you get a blue color, yes, you do have a gene, you have a possibility to get a breast cancer. If there is a red color, basically you don't have a gene. It's very simplified result of a very complex test. You can put the things together. As I told you, it's very interesting to put the proteins or molecules together to see how do they react. So you can use the DNA for this as well. You can make a DNA electronics. So that means because of the self-assembly, you can design particular chips. DNA itself is not conductive, but you can create layers of silver or copper on the DNA, so you can make a DNA electronics. You can even do the DNA computing, but don't think that you will be able to send your emails on the DNA computer anytime soon. You will not. But DNA computing could be very interesting also for biological systems, because, again, statistics, 1 kilogram of DNA has more information stored than in any computer that has ever been built. 1 kilo of DNA, and Joe Davis told you something about this. 10 trillions of DNA can fit onto 1 cubic centimeters, and 10 terabytes of data perform 10 trillion calculations in parallel computing. This would be enabled by use of a DNA in the computing and the use of this code in the computing as well. And of course, you know, DNA can be used as a novel material. It's still very expensive to make it, so it will be very expensive to have a shirt made of a DNA. But on a molecular scale, on a nanoscale, it can be used to make new materials where cell will grow easier. So you could basically use these DNA materials to grow organs one day, or to make stem cells feel a little bit happier, because stem cells don't like all the materials. So this work has been done also in creating DNA hydrogels. So all of this, you know, I started with a simple double helical structure, and I wanted to show you a little bit how important this is for our body, and then and for us as a living creatures as well. And then we went into the new area, which was enabled, which got created because a very creative person thought, well, that's an interesting structure, we can use that for something else. And now we have new areas developing. We have a synthetic biology, which is coming to life. We have a DNA computing. We can use the DNA for immobilization. We can use the DNA for aided crystallization. And I put lots of question marks here. Who knows what the next years are bringing to us? And I will finish with that. And I just wanted to tell you, you know, once when I do the talks, and sometimes I do the talks also for an audience where there are no scientists, and they ask me all the questions about nanotechnology and DNA and nanorobots and all that stuff. There are no nanorobots yet. Probably not in the near future. And you know, they think that people who are working in nanotechnology, they're some kind of special people, we have a special labs. But nanotechnology is basically a mix of different scientific disciplines. So we are chemists, physicists, engineers, biologists working together. So nanolabs look like any other lab. They're messy, they have a fume hoods, they have a test tubes, nice buildings, not nice buildings, and nano people look like any other people. Okay, so they usually don't pay attention until you tell them we are making a photo. Okay, thank you very much. I hope I inspired you a little bit to think about DNA. And if you have any questions, I will be happy to answer them. Thank you.